All right, it's 9 a.m. So we will start right now in order to avoid being late, even if some people will come. There are people coming almost every second now. So thank you for that. Thank you for being on time for this exciting day number 2 of ETPN 2020. I'd like to start maybe by thanking the people who came to the social event yesterday evening. Well, it's a lot of fun, I must say. And we enjoyed it a lot, even if it was a bit weird at the beginning, but we had some very cool and fun social interactions. So thank you for that. Today, so is day number 2, which is only the second, but we will have a third day tomorrow because the tab session will happen, the tab from the Health Tech tab, our mentoring service provided by the Nobel Project. But let's have a look first at the program of this day 2. I will try to share my screen and try to make it happen, not like yesterday. So, okay, I hope that you can see now the slide.
Fine.
Okay, and now maybe even in full screen. Woohoo! Okay, great. So what is the program today? Very rich program. We will start by a third keynote by Professor Joseph Kost from Ben-Gurion University of the Negev. And Spyros Anastasiadis, Professor Anastasiadis will do an introduction of Professor Kost, and I'm looking forward for this great talk. Then we will have a short session for dissemination of European projects in nanomedicine, like every year we do. For the last years, it was quite successful, and you will see that we have tried to make some kind of a mix because we have only 1 hour in this, let's say, shortened program of the virtual event, but quite a good diversity of projects. Then we will have a short break for 15 minutes, and then we'll come back for the second Pitch Me Up session. So we have 5 European projects and 5 speakers for the Pitch Me Up session number 2. It's going to last for 1 hour and 15 minutes. And then we will have the lunch break. So for 1 hour and a half. So let's say 12:30 until 2 p.m. And at 2 p.m., please stay for the great workshop that we have prepared together with New Deal and other sister H2020 projects. It will be a very practical workshop on translational issues for nanomedicine products with lots of different short sessions. And I mean, we've worked hard to make it very enjoyable and very interactive with you. So have a light lunch so, so you don't sleep after, because we need you to participate in this workshop, and you will see lots of great speakers and great panelists and great roundtables. So this is the program of today. I will stop sharing my screen. Okay, if I can. Oh, sorry. Am I sharing my screen? Yes, I'm still sharing it.
One way is to just close the file. That also better.
No, I need another option. I'm sorry. Sorry about that. Now it's shared?
Now it's Professor Kost.
Okay, but I just would like to stop sharing. Okay, I don't know why. Well, no big deal. Can you still see me?
Yeah.
Yes.
Okay. Sorry for that. We have some, let's say, some diversity in the behavior of Zoom every day. Why not? By the way, I will leave the floor now to start the meeting today for the keynote of Professor Kost. I will leave the floor to indeed to Spyros Anastasiadis, and please can you introduce Professor Kost to our dear attendees?
So good morning, everybody. It's good to have you all back today. It's really a pleasure for me to introduce Professor Joseph Kost, Yossi, from Ben-Gurion University of the Negev. He's a distinguished university distinguished professor and the senior advisor to the president on international research strategies of the Ben-Gurion. He holds the Abraham and Bessie Zaks Chair in Biomedical Engineering within the Chemical Engineering Department, and he has been the past Dean of the Faculty of Engineering Sciences at Ben-Gurion. He received his degrees, bachelor, master's, and doctorate in chemical engineering with specialization in biomedical engineering from Technion, the Israel Institute of Technology. And later on, he received also an MBA from the School of Management of Ben-Gurion. After his PhD, he went to the United States, first for a year to the University of Washington in Seattle, and then for 2 more years to the Department of Applied Biological Sciences of MIT and the Children's Hospital Medical Center of the Harvard Medical School. And in 1984, he went back to Israel and he started as a lecturer at the Department of Chemical Engineering of Ben-Gurion. He went through all the ranks and became also head of the department, dean of the Faculty of Engineering, and since 2016, distinguished professor. He has a number of distinctions internationally. He was elected foreign member of the United States National Academy of Engineering in 2007. The citation there says for discoveries that led to ultrasonic drug release and self-regulated drug delivery systems. He was elected a fellow of the American Institute of Medical and Biological Engineering in 1997, an honorary fellow of the Israel Institute of Chemical Engineers, a member of the Controlled Release Society College of Fellows in 2014, a foreign member of the Polish Academy of Engineers in 2014, a member of the Israeli Academy of Sciences and Humanities in 2015, and in 2016 he was awarded the Israeli chapter of the Controlled Release Society Award for Outstanding Achievement in Drug Delivery, in recognition, of course, of his pioneering work in the fields of ultrasound-based drug delivery systems. His research interests, as it's obvious, are in the field of biomedical engineering, nanotechnology, biomaterial science, controlled drug release, gene therapy, and ultrasound. And we're very happy to have him with us to talk to us about nanoscale systems and strategies for targeted delivery of drugs and genes. Professor Kost, we would have been happy to welcome you live in Heraklion, but we are happy that we have the opportunity to listen to you today.
Yeah, very much regret not being able to be there and see you in person. Thank you very much for this kind introduction.
Okay, sorry for that. Thank you for the introduction. Just a word, I forgot to say that indeed you can ask your question anytime in the Q&A section in the bottom menu for those who are not familiar, bottom menu of your Zoom tool. And you can also ask it by raising your hand or directly typing any question in the chat. Sorry for that, but that's very important because we want questions for Professor Kost. So now I will broadcast your lecture and Professor Kost will be here for the Q&A. Enjoy your lecture.
Good morning. Very much regret I cannot meet you in person in Crete. I would like to start thanking Professor Spiros Anastasiadis and the organizing committee for inviting me. The topic of my talk is nanoscale systems and strategies for targeted delivery of drugs and genes. The ultimate goal is to administer therapies at the right time and the right dose and the right place in the body with specificity and efficiency. Focusing on the right place, if we go back to 20 or 30 years back, we can see that the therapies focusing, targeting, moved from macro to micro to nano. They actually moved from invasive approaches to much less invasive or even non-invasive. In terms of targeting, that's the place I refer to, moving from large targets like the organ targeting to micro and nano to the intracellular targeting. What I would like to do today is to present Nano-Scale, to present that Nano-Scale offers unique strategies to target therapies at higher specificity and efficiency. And I'll present 4 strategies. One is the route of administration, enhanced permeability and retention, what's called EPR, ultrasound, and ligands. So let's start with the first, which is the route of administration. Whenever we refer to route administration, we can talk about delivery to the skin, topical, could be intratumoral, could be pulmonary, ocular, and there are many, many others. I would like to present an example from our lab, which would be pulmonary targeting. What you see here is a picture, a photo of a premature infant. And premature infant, one of the issues are lung diseases. And actually one leading is that these premature infants, since they lack the surfactants in their lungs, whenever they are born, their lungs cannot inflate. And since they cannot inflate, they cannot breathe. So the approach these days, and this is a report for the last, I would say, maybe 20 or more years, is to administer to their lungs through these intubation surfactants. So there is a work in different groups on improving the surfactants. And we were collaborating with a group in the Cincinnati Children's Hospital that they are developing a novel surfactant, but still the issue is that these premature babies has to be incubated for several days to deliver these surfactants. And our approach was to study if we can deliver the surfactant and other surfactant, but the surfactant that was delivered in Cincinnati with a targeted approach that would alleviate the need of long or frequent incubation. So the approach was to prepare nanoparticles. The nanoparticles that would incorporate this surfactant, which is the SPD, developed in Cincinnati. We for that used well-known biocompatible polymer, FDA approved, which is presented here, composed of lactic and glycolic acids, endogenous acids, so the biodegradable polymer is degraded to endogenous molecules. This is the surfactant incorporated into nanospheres. You can see this SEM image of these PLGA nanoparticles, and you can see the size in that nano range are these particles. And the idea was, as I presented, to deliver them and have the SPD and other surfactant released into the lung. We did present just one result, which is an in vivo result. Again, this is the nanoparticles, depicted here quite large, delivered to the mouse lung, and you can see the concentration, or this is this SPD concentration in the lungs at 2 doses, and this is untreated, as you see, meaning no nanoparticles. And you see the high concentration of the SPD of the surfactant. This is 2 days after intratracheal injection. It was recently published in the International Journal of Pharmaceutics. So this was kind of the first strategy that I presented. The next 3 other strategies are in the field of gene delivery. So before going to these strategies, I would like to briefly introduce what's gene delivery. So what is gene therapy? Gene therapy is when we deliver the gene, could be a DNA or RNA, we introduce it into patient to treat genetic diseases. This plasmid usually contain functioning nucleic acid sequence to correct the effect of disease-causing mutation. So the objective here is to take the DNA or RNA here using a carrier, could be viral or non-viral, get it, inject it to the body, get it all the way to the cell, and if that's DNA, to the nucleus, if this is an RNA, to the cytosol, to get a secreted protein or actually a silencing of some proteins that are released in high concentration. I mentioned carriers being viral and non-viral. Viral, this is an example of viral, the way it is approached is that you remove part of the virus DNA and incorporate into this his DNA the sequence of interest. So in the viral approach, there is a limit on the size. There are some upscaling problems, but the main issue, especially now these days with the COVID-19 epidemic, safety is for sure an issue. So we and many other groups are developing a non-viral approaches. There are advantages, the main being the safety and unlimited size of the DNA or RNA which we can deliver. The disadvantage is the transfection efficiency. Targeting and getting the high concentration of the DNA to the cell or to the specific cell or to the nucleus. The non-viral approach is based on carriers. I'll focus on polymeric, could be liposomal, but I'll focus on polymeric carriers. These carriers could be positively charged since the plasmid, DNA, RNA are negatively charged. Now, the negative charge, since the cell surface is also negatively charged, is a disadvantage or an issue. So what we do, whenever we have this, another issue is the size, because this negative charges repel, and this DNA is quite large to penetrate the cell membrane. If you use these carriers and we kind of bring these two together, we have a self-assembly process, and the large size condenses to small nanoparticles in that nano size. And if the ratio of the positive charged molecules versus the negative charged molecules is higher than 1, we can even get a surface which is positively charged, which is advantage in terms of cellular uptake. Let me present to you the process of this condensation. What happens whenever we start adding the cationic, the positively charged carrier? So we start from a naked DNA. You can see this is a macaroni structure. And now I'll start adding the positively charged carrier. And you can see maybe the starting of a condensation. Now the ratio here, which we called N to P, which is the ratio between the molar number of cationic groups versus the anionic. So N being the cationic, the amine, P being the phosphate, the negative. So this is N to P equal half. See what happens whenever I increase the concentration, we have more carriers, N2P, and you can see very nicely how this nanoparticle will actually condense this DNA, but still not a full condensation. So if we continue and increase the number of N2P, the ratio, you can see very nice nanoparticles in a size that can, by endocytosis, internalize into the cells. So, but still, the process of getting these nanoparticles to the nucleus of the cytoplasm is not without barriers or without hurdles. So let me present to you the hurdles. So this is, again, the DNA, which is negatively charged, our carrier positively charged, self-assembled to nanoparticles, and the first barrier would be internationalization into the cell. Once it gets into the cell, there would be, because the body regards it as a foreign body, it would be engulfed into a vesicle that is called endosome. And if it stays in the endosome, that endosome would turn to lysosome and will degrade this nanoparticle. So we have to make sure that there is a release of the nanoparticle from the endosome. And now we have trafficking towards the nucleus. And another barrier is decomplexation, because in order to get this DNA or RNA biologically active, it has to decomplex from the carrier. So as you can see, not an easy process or targeting process. So let's go back to targeting strategies. I presented the first to be the route of administration. I would like to move now to the second, which is based on a phenomenon which is called enhanced permeability and retention, EPR. And what's that EPR? So this is a schematic presentation of a vasculature in a normal tissue region versus the vasculature in the area of a tumor, cancerous tumor. You can see that in this normal tissue, these are the endothelial cells, these are tight junction, and these are very, very nice tight junctions with very, very small openings in the range of nanometers. When we move to the vasculature, the vessels in the area of the tumor, since when the tumor growth needs fast supplement of nutrition, there is a fast growth of the blood vessel or the vasculature toward the tumor. It's a phenomenon called angiogenesis. And due to this very fast growth, the vasculature of the blood vessels are leaky. They are not perfect. You can see this fenestration, there are openings. And in tumor capillaries, this could be in the range of 100 to even 1 micron. So if we have nanoparticles in that range, which is higher than, let's say, 100 nanometers or 200 nanometers, they cannot permeate through the blood wall in the normal tissue region. But once they get into this tumor area, they actually can leak out. They can penetrate the wall through these holes and accumulate in that tumor tissue region. So actually what we have is kind of a passive targeting. We inject IV and get accumulation in the tumor area. So let's see an example. This is a PhD work of one of my students, Elise Lewis, and the approach here Well, the goal was to shut a natural pump of cells, which is causing a phenomena which is called MDR, multidrug resistance. And what it is, in these cancerous cells, we have high concentration of P-glycoprotein, and that P-glycoprotein controls this function of a pump that whenever there is a cytotoxic drug, actually any other drug, but whenever there is a cytotoxic drug that enters the cells and we give this chemo to cause apoptosis called dead cell, what happens is that there is a pump that actually pushes that cytotoxic drug out. And we are not getting the expected effect of this chemotherapy. Now, the molecule that controls this pump is P-glycoprotein and would like to silence this function. And to silence this function, we can do that by delivering SI small interference RNA, which is small interference PGP, which would actually shut the pump that is pushing the chemo out of the cell. So the carrier that we developed for this approach is based on starch. Starch is composed of two polysaccharides, amylose, which is a linear, and amylopectin, which is a branched polymer. The starch is biocompatible, non-toxic, stable, and biodegradable, so very good candidate to be used for such application. The problem that we have to address is the starch and many other polysaccharides are non-charged. So we have to do this modification, and we modified the starch with quaternary group, which is the amine group, which has this, which is actually positively charged, so we can get what we call quaternized starch, which is positively charged. So here we have this quaternized starch. Once we get it with that siRNA, we get a self-assembly of this complex, a nanocomplex, that would be internalized by the cell to cause some release from the endosome and release of this siRNA, starting this siRNA machinery to silence the PGP, silence the pump. You can see here the size of these complexes. Here I'm showing really the silencing, gene silencing using that siRNA that is caused by these complexes. This is the SI-PGP complex, a different N-to-P ratio. So if this is, and these are untreated cells, you can see the PGP expression, which we call 100%. And if we deliver these nanoparticles, we can see the silencing from 100 to 70 almost, and here to 50 at the ratio of 1 to P2. These are the carriers, and you can see the effect on silencing. But I wanted to show you the targeting. If you remember, this is the EPR effect, and what we did here, and that's the result that I'll be presenting, is this is an in vivo tumor uptake. So what we have a mouse that was inoculated with 4 different cancerous tumors. And now we deliver these nanoparticles, and since this is a fluorescent label, we can follow their fate after the intravenous systemic administration, as I said, labeled complexes. And you can see this accumulation, very nice accumulation in the tumor. Yes, there is also accumulation in the liver, but definitely accumulation in this tumor, and this is due to this EPR effect. So again, the strategy is to use the EPR to target tumors. And this is another kind of results of that experiments, systemic tumor uptake, showing the really uptake of these Q-starch enzyme complexes because of the size, as I said, EPR versus control, which was just a dextrose, and you can see now just the siRNA injected in dextrose, no uptake in the different post-injections. The next targeting strategy is ultrasound, the use of ultrasound. So what's ultrasound? Ultrasound is sound wave at a frequency higher than 20 kilohertz. So we have this is the audible sound, and this range is the ultrasound. There are numerous applications of ultrasound in medicine, in the low frequency dentistry, the mid frequency therapeutic, and high frequency diagnostic. Therapeutic, mainly physiotherapy to hit muscles and relieve pains. Now, one of the phenomena that we take advantage of ultrasound is cavitation, which is the formation and oscillation and collapse of gaseous bubbles. So in the liquid that is exposed to ultrasound, there is a dissolved gas. Once we expose the liquid to ultrasound, that dissolved gas generates bubbles, and these bubbles rotate. And you can see these bubbles here, and this is the bubbles that rotate. And actually you see this, the sound pressure, these are the bubbles. And in cavitation, we distinguish between stable versus inertial cavitation. Stable cavitation is whenever this bubble rotates between the two distance diameter. But when we increase the intensity, the bubble starts to rotate and expand in size, as you can see here, to the point that they rupture and collapse. In this collapse mode, we generate shock waves. You can see here these microjets, which generate stress, microstreaming, and effects we take advantage of. So now the next example to present the effect of ultrasound as a targeting strategy would be a PhD work of Renat Lifshitz, was in collaboration with Professor Chesidi and Dr. Avni from Sheba Medical Center. And in psoriasis, it's well known that in these psoriatic cells, there is high concentration, high expression of two proteins, interleukin-17 and interleukin-22. It is also known that in order to silence, reduce the concentration of these two proteins, you have to deliver mere microRNA, it's called miR-197, specific RNA, that would cause silencing of these two proteins. So our approach was to try and deliver this miR-197 to psoriatic cells. Now, if we look into the skin, and this is a cross-section of the skin, you can see different layers. The top layer is the stratum corneum, which is the barrier to transport of the epidermis. And we have this, beneath the dermis, we have the basal layer. And where this is the layer that we would like actually to target, the layer that we would like to target these psoriatic cells. This is a psoriatic skin cross-section. You can see the psoriatic lesion on top of the skin. So our goal is to try and get our nanoparticles all the way through this lesion to the target, which are the basal cells. Now, the top layer of the skin, as well as the lesion, is impermeable. As before, the carriers which we'll use is the quaternary starch, which is a cationic, but now we complex it with the specific miR-187, get these nanocomplexes, but for the targeting, the approach is to use ultrasound, and use that ultrasound as a pre-treatment before we apply these complexes. So the complexes will never be exposed to ultrasound that might affect these complexes. So here, this is schematic presentation. We apply the ultrasound to the top of the skin in that lesion area. And this is a very brief application. Once we are done with the application, we remove the ultrasound, we put on these nanoparticles, and we'll follow their fate if they'll get into the cell, internalize, and get into this showing a silencing effect. So this is kind of characterization of these complexes. We look for the right N to P ratio, and you can see that the naked RNA being negative would run in this gel to the positive, but once we increase and make the complex, get to the point that it stays and is not running in the gel. You can see the AFM. You can see, in this case, the hydrodynamic, the ratio used was N to P2, the hydrodynamic ratio at this different mesh. The hydrodynamic radius is, for N to P, let's say 2 is about 25, so we're talking about nanoparticles of 50 nanometers in size. And this is the zeta potential going from 0, increasing more carrier to the point that we have positive zeta potential. Now, here we go and study the transport of our nanoparticles through this psoriatic skin. So this is psoriatic human skin that was transplanted onto mice. As you can see here, this is the cross-section of the skin. This is the top layer, the area of the lesion, and these are the stratum corneum, epidermis, dermis. Our microRNA now is labeled by Cy5, it's red. So actually we follow the red and we see that without ultrasound application, all the complexes stay on top of the skin. But if we do this short pre-treatment with ultrasound, you can see the very effective, efficient transport of the complexes down to the layers of interest. So again, we are discussing strategies for targeting. And here we use ultrasound to target these complexes to the basal layer into these psoriatic cells. As I mentioned, in these psoriatic cells, there are two proteins which are highly expressed, high concentration, although show the data for the IL-22. And again, this is in vivo biochemical evaluation because we're looking for a biochemical effect after 1 week into this psoriatic human skin transplanted onto mice. And you can see no penetration without ultrasound. And you see this is kind of a strong green, high concentration of this IL-22, which is labeled in green. With short ultrasound application, you can see again the penetration, but the additional result that I would like to present is the difference between this high concentration and here the silencing effect of our nanoparticles. The final kind of step was the in vivo, evaluated in vivo, and that was done again on the mice transplanted with human psoriatic skin. And what we have here are 4 groups. We have a psoriatic skin, healthy skin, psoriatic skin, and this is after application of placebo nanoparticles with microRNA that has a different sequence, not the Mir197. And this is our experimental group that received the complexes. So what we did, we took these 4 groups, treated with ultrasound, and applied the nanoparticles for these 2, and this is no treatment, and this is healthy, no treatment. 1 week after treatment, slides of the skin taken from these mice were taken to independent dermatologists to evaluate 5 psoriatic marks: epidermal thickness and shape, elongated nuclei, neutrophils, and multiple blood vessels. Now, based on these 5 scores, the dermatologist gives a total visual score, which is for psoriatic skin above 9, for healthy skin about 4. This is the group which received the RNA, the scrambled RNA, which is a control. We're not expecting any effect and there is no significant difference. This is an experimental group after 1 week. So you can see improvement in the visual scoring going from more than 9 to 6 after 1 week. And this is a kind of study that we continue. So again, the focus was to present to you the strategy of using ultrasound to target these nanoparticles, again, taking advantage of the nanosize. The future, how we envision that, we envision if there is a psoriatic lesion, very brief application of ultrasound, followed by a patch that placed in that area and could be left there for night, or depending on the severity, and kind of relieve the psoriatic lesion. I mentioned ultrasound. The ultrasound we started with ultrasound, which is kind of well known in any biological lab, but we developed that. This is an ultrasound we developed for transdermal application, just for delivery, which got an FDA approval for delivery of lidocaine. And more recently, we're now collaborating with an Israeli company that is developing totally non-invasive application of ultrasound, and we evaluate this application for the targeted delivery but these devices are already FDA approved for the treatment of uterine fibroids. We use ablation, pain from metastatic bone cancer, essential. The big advantage is that non-invasively we can target the ultrasound to any specific area in the body. Now the last strategy, so I presented the route of administration as permeable ultrasound. The last strategy that I would like to present are the ligands. I'll present actually combine targeting using both ultrasound and ligands. So first, let's start with ligands. What are these ligands? Ligands are molecules that bind particular cellular receptors, can be attached to a nanoparticle to actively target cells expressing these receptors. So if we have a cell, let's say cancerous cells, that has specific receptors on its surface, if we can find a ligand, a molecule, that would bind preferably to these receptors, we can now attach this ligand to our delivery system and deliver the drug of interest to these cancerous cells. So again, let's go to an example. Here I'll be presenting a use of a different polysaccharide, Pectic Galactan, for targeted gene therapy for glioma brain tumors. This work is in collaboration with Professor Henry from Johns Hopkins. Work was done, again, PhD study by Nitsa Boron. So this is brain cancer, glioma, survival of months. You can see here now these glioma cells are expressing highly molecule which is called Galactan-3. So we have specific receptors at high concentration on the glioma cells. So we thought, how could we take advantage of these specific receptors on the glioma cells? And we this is well known, that Galactan is highly specific to Galactan-3. There are specific interaction and high interaction between Galactan and Galactan-3. So a Galactan could be used as a ligand to target cells that express highly Galactan-3. Now, looking to Galactan, there is a polysaccharide, natural polysaccharide, which has on its backbone high concentration of this Galactan group. And this is pectin. Pectin is, as I said, natural polysaccharide. In this pectin structure, we have early areas where there is a high concentration of Galactan. So we cut this pectin to this high concentration polysaccharide with Galactan groups. This will be the Pectic Galactan. Again, Pectic Galactan is not charged. So we did very similar chemistry, modifying it to be positively charged. So we have now Q-quaternized Pectic Galactan. And once we have this modified Pectic Galactan, we can do this again, the self-assembly with the plasmid DNA. Since we are interested in delivering this DNA to cancer cells, we will take a plasmid DNA that is causing cell apoptosis. It has to get to the nucleus, and that would cause cell apoptosis. So again, Galactan being our ligand, this is the receptor, generate these nanoparticles with the ligands, attach them to these glioma cells, high concentration. So let's see some data. This is the cellular part in vitro. The plasmid is labeled in blue, the carrier in green. This is the cell. Actually, the membrane is labeled in red, so you can see them all here. The membrane, this is the cell. You can see this kind of green, which is the carrier. There is some blue, but if you look, this is the blue. It is in the area of the green, but this is a merge. But I'll show later. The nucleus here is in the center, and we do not see much in the nucleus. We do see cell internalization of these nanoparticles. So we have a cellular uptake of 75%, but very low nuclear uptake. So we thought, okay, let's try our approach using ultrasound. Maybe we can, by the ultrasound, overcome some of these transport barriers. So what I'll show here in the upper figures, this is without ultrasound application, and these are the cells. This is free DNA. I'm looking for the DNA, which is again labeled in blue, carrier in red, and the membrane carrier in green. So this is free DNA. This is the free carrier, as I say, in green, and you see the internalization, but none in the nucleus. And this is the complex. And again, the complex, you see green and blue, but none in the nucleus. Let's compare it to this short application ultrasound. This is 20-second ultrasound, low intensity, applied 24 hours post-complex exposure, and cells were fixed for 48 hours post-exposure. So we're looking now 48 hours post-exposure. And again, this is with ultrasound, following ultrasound. This is free DNA blue, we see some. This is free carrier green, we see some. Remember, the membrane is red. And see the very significant effect on the nuclear uptake. The DNA is blue. These are the nucleus, and see how blue these are. So again, using ultrasound, we were able to target now our nanoparticles to intracellular organelles. Now let's see some in vivo experiments, and the in vivo was done by NITSA in Henry Brem's laboratory, Johns Hopkins. I would go to this setup, but we made sure that the ultrasound intensities are safe, the concentration of nanoparticles are safe. And what we found another advantage of the approach. So this is the tumor. And this experiment, actually, the nanoparticles were injected intratumoral. And you can see this is the site of the injection. And you can see this is the tumor, relatively low distribution. But this is the injection followed ultrasound application. Again, low intensity and short exposure, but you can see the significant effect on the high distribution. And also, now, as I mentioned before, this is what I'm showing again, this is the same legend. You can see this carrier cell nucleus And you can see here the significant effect in this term passive, and you can see this DNA. Please notice that DNA now is labeled in red. The cell nucleus is blue, and no ultrasound cells, the nucleus is in blue. Once we do this short application of blue, you can see the nucleus start to be red. The pink, the red. And you can see these two effects on diffusion and also on nuclear uptake. So to kind of summarize before acknowledging what I presented are 4 strategies that present the advantage of the nanoscale in terms of targeting therapies to target of interest. I would like to acknowledge my students, the funding sources, and my collaborators. And thank you from all the group for listening. Thank you.
And we are back. Thanks a lot, Professor. Excellent presentation, very wide, and it's a great pleasure to benefit from your experience. So first, I think we have a question here, and yes. So we have a question from Raymond Schifflers from UMC Utrecht. So 50 nanometers particles are quite small in this essential. Is this essential for the skin permeation in combination with ultrasound?
Yes, that's what I was presenting. Without ultrasound, the nanoparticles don't permeate the skin or the psoriatic lesion. Please remember that we are now trying to permeate the psoriatic lesion, which permeability is different. But the 50 nanometers are not permeating the stratum corneum without the effect of ultrasound.
Thanks a lot. I have a question. What is the level? I mean, what would be the next step of development for your treatment against psoriasis? Do you plan a clinical development very soon? At which step are you? Would you like to spin off a company for this specific application? Because I mean, the data are quite impressive.
Well, all points you mentioned are of interest. We're continuing, and we would like to show a more significant effect. After a longer time, we showed the reduction. But now really to get it into clinical, we have to address all the safety. As I mentioned, the ultrasound that we are using was already approved by the FDA for other applications. So the ultrasound is not so much a safety or an issue from the regulatory point of view. And the other thing is that we are not really exposing the nanoparticles to ultrasound, so there are no issues that maybe whenever expose them, there are some other effects, but still this is something which we'll have to show. So yes, very much we are interested moving to larger animal. The problem is having this animal model, to do these studies on not on a human implant, but really on doing it on a better maybe clinical model or preclinical model. But we'll be very much interested in having somebody to help us move it forward.
Apply to the health tech tab. By the way, we have a new question from Andreas Aslund asking, it does not look like transcranial ultrasound was used during glioma treatment. Why is this?
At that time, we did this experiment in Henry's lab, and these were the ultrasound that were available to us. Now we are collaborating with Insightec, and we can repeat this experiment with a totally non-invasive ultrasound that Insightec is developing. Actually, they have the helm with a lot of transducer that they can target the brain tissues totally non-invasively. This is something which we are now in the process of doing.
Thank you. Now we have a new question from Iraida Loinas from CDTech. And Iraida is asking, how important is the functional group trimethylamine you selected on the transfection efficacy?
The importance of that group is to turn the polysaccharide to be cationic, so we'll have a self-assembly with DNA or RNA. In terms of its biological activity, I don't think it would have an effect, but that's the way to complex, get the right size and get the nanoparticles to the target of interest.
Okay, a new question for the ultrasound, which is, how do you measure the needed time of ultrasound for different purposes?
We evaluate, in ultrasound, there are several parameters that you can really control. You can change the frequency, you can change the intensity, you can change the probe of the ultrasound having different shapes, and definitely you can change the time. Overall, one of the parameters would be the total energy. So you can maybe lower the intensity, extend the time. We are looking into times that would be reasonable for the application, but this is something which we are evaluating. So based on the energy that is needed, and we evaluate the time, preferably we use it for short times. Now, in regard to the skin, and actually what we have kind of a feedback control. So when we expose the skin to ultrasound, we are interested in increasing its permeability. We follow online the electrical conductance. And as the skin permeability or the lesion permeability increases, the conductivity increases. And there is a shape of that change in conductivity, which is a sigmoidal shape. So actually we know exactly when we are halfway and we can stop the ultrasound at the point that we have the high permeability without causing pain to the patient. So there are ways to address by some ways to follow the change in conductivity.
Very clear. New question from Speranta Tanasescu from Bucharest, and Speranta is asking, have you planned on a thermodynamic study of the interaction at the bio-nano interface by BITC, for example?
No, and I'll be very much interested to discuss and see how we could do that, but no.
Okay, this is the purpose of this meeting, is to create collaborations. Finally, and I think it will be our last questions, from Nicolas Carvou, thanking you for the great lecture first, and say, for the enhanced EPR effect, nanocarriers for the enhanced EPR nanocarriers, have you ever thought of combined magnetic hyperthermia treatment?
Yeah, I thought, but nothing was done. So the question is yes and no. Again, this is something which is of interest, and there is a use of ultrasound as an ablation trigger. We are trying to keep the temperature without causing that ablating effect, but we are developing also some different microbubbles to enhance the thermal effect. So this is something which is definitely of interest to us.
Okay, great.
Spiros has a question.
May I? It's
Please, come on.
When you discuss the complexation of the cationic polymers or the starch with the plasmid, the negatively charged plasmid, there it seems that it's just an aggregation because of charge, right?
That's correct. Ionic interaction.
Yeah. So is the size distribution, how important is the size distribution? Because there you probably have a broad size distribution because it's not like thermodynamic micelle or something. It's a different kind of application.
I showed the size distribution. The size distribution is not depending really on the application. If the size distribution is very high, we could lower the size distribution using different separation approaches. The size distribution is a factor, but it's not a huge issue at this point.
Okay, thank you.
And last but not least, I forgot about the question of Ruth. How may I? Ruth has a question on the EPR effect. So she said, thank you for the nice overview of various targeting strategies. You mentioned the EPR effect for tumor targeting. This effect is known to often be overrated in animal models. Do you have any strategies to find the patient groups that have a significant EPR for this targeting approach?
It's an interesting question. Some people don't believe at all at the EPR effect. As far as I know, one of the first approved drugs, which was the liposomal doxil, is based on the EPR effect and seems to be working pretty well. I'm not a physician to address the clinical studies, so maybe somebody in the audience would be a better one to address it.
Well, Professor Kost, once again, thank you very much. And I would like to add the same comment that I've made for Patrick Bamrauf yesterday. You said yes at the first email upon invitation of Spiros, and then you said yes to the whole crappy email that I've sent, like, I'm sorry, but we need to move, we need to move, we need to move, and we need to go fully virtual. And every time you were the first to answer yes, very positively, very positive move. So that's precious when you organize an event. So thank you for that. And you can.
I was so much looking to come to create and meet the group there. And
you have an open invitation. You have an open
invitation. But once again,
thank you so much. I enjoyed it yesterday, and I'm sure it would be an exciting day today. Thank you.
Thanks a lot, Professor. Thanks again. So.